what is protein function? Recently, like a lot of different people have tried to answer this question, and there have been a lot of different types of answers. So I just want to start with defining what is a protein's function. And I'm going to be using this protein fuss kind of throughout my presentation as an example, primarily because it's a really well-studied protein and a lot of effort has gone into annotating it. And some of the functions I'm talking about are like DNA or RNA binding, transcription regulation, RNA splicing transport, DNA damage response, that kind of thing, like what the protein does kind of as a whole. And also recently, protein language models have popped up, and they've been able to make a lot of really great predictions about proteins in a lot of different fields. But today I'm going to be talking specifically about unsupervised protein language models, which are trained by only seeing protein sequences. They don't see like annotations from experiments or domains or anything like that. And once the model is trained, we can give it a whole protein sequence, and it gives us a protein embedding, which is simply a numerical representation of the input protein sequence. And then using those embeddings, we can, without further training, do some really interesting stuff like predict protein function better than BLAST. And I think that shows that they're learning something a little bit more than just sequence homology. And we can also visualize protein embeddings to kind of get ideas of what's going on in the protein embedding space using something like UMAP and then a scatter plot to look at it in 2 dimensions. And since it's unsupervised, we can actually do some really interesting stuff like analyzing rare proteins or novel proteins. And one of the reasons we can do this is because it's unsupervised, so we don't need a lot of examples to train a supervised model on those rare things. And just again, I can do all of this just from looking at the protein sequences alone. And if we look at protein function at a higher resolution, as many people probably know, they have independently functioning regions like domains that perform a very specific function. Like here we have an RNA recognition motif that is like helps with RNA binding, of course. And then even some of the disordered regions like the tail at the end have important functions also, like localization to different parts of the cell. And we might be able to imagine how all of these different independently functioning regions kind of work together to perform those whole protein functions, like as a reminder, like DNA and RNA binding, that kind of stuff. So what I've been working on recently is looking at if we can use embeddings from an unsupervised model to study like these functional regions within proteins instead of at the whole protein level. And yes, we have kind of figured out a way to look at this kind of stuff. So just to go through this, like if we give the FUS amino acid sequence to a protein language model like T5 that was trained unsupervised, it gives us a protein embedding. And here we have like a per residue embedding where each like column in the embedding corresponds to an amino acid in the protein sequence, and each row represents the high dimensional space of the protein language model. And if we actually just like look at the heat map of the embedding, we can actually see there's kind of like distinct portions along the protein sequence that happen to line up with annotations from the literature. So what we did next was we used change point analysis to define the portion. Sure. I don't know.
You can ask that at the end.
Okay. Okay, so we use a change point analysis, which is a simple algorithm. It doesn't have any trainable parameters, and we use it to define or like cut out those segments in the embedding that are distinct from one another. And then we average pool, and we get these 1 by 1024 embeddings that correspond to the segments that we can visually see in the whole protein embedding. And this is again, done without any trainable parameters. So it's also an unsupervised process. So we're kind of like maintaining the unsupervisedness as we go through this. And then what we wanted to do is to check whether the segment embeddings are actually capturing something meaningful, or if I just randomly cut it up. So we took the human proteome and I generated those segment embeddings. And when we take segments that overlap with the top 20 most commonly occurring domains, and we make a UMAP out of them, we actually see that there are strong associations with the segment embeddings and protein functions. So these segment embeddings are associated with protein function. And something we wanted to do is, instead of having this kind of like ginormous visualization, we wanted to know if there's a way that we can visualize these functional regions in proteins more similar to how they're represented in the molecular biology literature. So what we did was we took the segment embeddings and we reduced them to 3 dimensions instead of 2. And then we scaled those values so they can be interpreted as RGB colors. So in the computer, you have your 3 kind of like primaries, like instead of red, yellow, and blue, you have RG and B, where each one of those numbers represents how much red, how much green, and how much blue. And we can use those to map to a very specific color in the color range on computers. And then we just scaled it up to represent the actual space of the protein that the segment is associated with. So we can use these colors to look at like similarities and differences in the high dimensional space of the segment embeddings as a way to just like easily visualize the embeddings. And as an example, I took these RNA binding proteins, including my example protein FUS, and we took the segments, we generated the colors, and when we compare them to annotations from the literature, which are shown above in gray, we actually see that the colors are associated with annotations from the literature, and also we looked and checked that the embeddings are also closely associated with colors, so it's accurately representing what's going on in the high dimensional space. And I just want to take a moment to emphasize that this is also done completely unsupervised, and the colors are also unsupervised. There's zero shot in a sense where we don't train parameters, so we're actually just able to see what's going on with the embedding. And to just quickly recap, we have found a way to like define and visualize these functional regions and proteins from the protein language model embeddings. And I actually have like a little tool if you want to see proteins from the human proteome. You can check that out, and it runs on Colab, so you don't actually have to install anything. And it generates something that looks like this. Okay, thank you to my lab members in Melissa's lab and our collaborators that have helped.